Hi everyone. So I'm going to talk about biotechnology in this lecture. So this is a really cool lecture because you guys are going to learn about how everything you've been learning about in regards to DNA and RNA and protein is actually used in industry and medicine every day. So this picture here shows an E. coli bacteria that's been genetically modified to produce an important human immune system protein. And this is going to become important in medicine when we talk about it later. Now, we can transform, the word is called transform. We'll talk about what that means later, bacteria with different genes. And remember from the last lecture that when you take the host's DNA and you insert a different gene in it, we call that recombinant DNA because it has a combination of DNA from different organisms. We can transfer that to the bacteria, and then that bacteria can make different products for you. So for example, you can get bacteria to make genes for you that code for different proteins that help with plant cells, help with pest resistance, weed resistance. You can work to create enzymes in bacteria that will degrade different toxic chemicals. The enzymes, and remember again, enzymes are proteins, amylase, cellulase, and other enzymes are used in clothes manufacturing to break down different types of stains. And you can create the different types of human growth hormones that will treat stunted growth, different issues with growth in humans. So we're going to talk about biotechnology, and in this lecture we're going to talk about different types of experiments that are done in biotechnology. So we're going to briefly talk about DNA sequencing, what DNA probes are, the experiments they're used in, DNA fingerprinting, and this is exactly what it means. How do we use DNA to figure out what an organism is, which human is that specific person? And we are going to talk about DNA cloning. DNA cloning is a very popular biotechnology experiment that's used every single day in companies to create different things. And we will talk about that. So I'm going to start off with the definition of biotechnology. So biotechnology is basically using microbes or cells or cell components to create any useful product. So it's using the biology of cells in technology to create useful things and solve practical problems. The biotechnology field is huge, so we use microbes and cells to create things for us in medicine, different types of medications that we use every single day, such as insulin, were the product of bacterial biotechnology experiments. We use them in industry. We create different things for plants, for washing, for creating plants that don't have seeds, seedless plants. So that's biotechnology. And then forensics. How do you identify who is the crime in a murder case? How do you use that DNA? And components of biotechnology, so different experiments that are used in biotechnology are things that help you identify DNA. And this is where we're going to talk about DNA sequencing, DNA probes, and fingerprinting. And we are going to learn about what DNA cloning is. So DNA identification technologies. So there's different ways that we can identify DNA, different experiments that can be done. So we're going to talk about DNA sequencing, DNA probes, which are these molecules that are used in different experiments. We're going to talk about an experiment called FISH and DNA microarrays. And then DNA fingerprinting, we're going to talk about PCR and restriction fragment length polymorphisms. So to start out with DNA sequencing is exactly what the name says. So it's basically when you take a DNA molecule and you figure out the sequence or the order of base pairs in DNA. Remember that DNA is made up of adenines, thymines, guanines, and cytosines, which we abbreviate ATCG. So this is when you take the DNA of an organism, whether that be a human or a bacteria, and you basically stick it in a machine, and the machine tells you that the order of this organism's DNA is like GGCATT. Now, you may wonder, why is this important? And especially with the coronavirus, this is becoming important when you've heard about different companies sequencing the coronavirus genome. They're actually sequencing RNA because coronavirus is an RNA molecule, but you can sequence RNA. And the reason why it's important to do sequencing is when you get the different order of base pairs, you can figure out what genes different organisms have by looking at these base pairs. You can figure out what they're related to. So for example, in the case of coronavirus, if we sequence its genetic molecule, RNA specifically, we can see, is it more related to the SARS virus? Is it more related to another common cold virus? This gives us an idea. Same thing with humans and bacteria. We get an idea and a better understanding of what are they more closely related to so that we can better understand an organism. You can look at if an organism has certain genes. Does it have the gene for flagella so that it can move? And the Human Genome Project, this was done about 20 years ago, and it's when scientists sequenced the entire human genome. So the reason why this is important is because we can maybe use, for example, cancer patients and people who do not have cancer and look at the differences in genes, what genes are targeted with different diseases. So the human genome is about 3 billion base pairs. It's about 25,000 genes. There's a lot of genes in there, or DNA, and as scientists we call it junk DNA, and really we call it junk DNA because I think we really haven't discovered what it codes for. I think a lot of it may be useless, but with time there's a lot still to be figured out about these new genes. So that's the importance of DNA sequencing. And whenever you have an outbreak, such as what we have right now, and it's a very new organism, the first thing you want to do is sequence its genetic information, whether that's its DNA or its RNA, so you can get a better understanding of it. And this can also be used for medicine to target different potential genes that an organism has. And when the human genome was sequenced, it was a really big achievement in science. So we have many other organisms that have been sequenced. Okay, so that's DNA sequencing. DNA probes. I want to talk about what this word probe means because we use it a lot in different experiments. So DNA probes are complementary sequence molecules that are complementary to DNA. And so if let's say there's a specific molecule in DNA you want, the specific gene in a DNA molecule that you want to study. So you have the whole genome, but you just want to focus. Let's say you have an organism that is motile and it causes infection like a bacteria by being motile. So you want to study that gene more. So what you would do if you want to focus on that gene, you create a molecule that's complementary to that gene, and we call that a probe. So if the gene you're interested in, the sequence of it is ACC, GGCC, you can create a molecule that's complementary to it, and therefore it will bind to it, and you can do a lot of different experiments with it. So maybe one thing is you can add a fluorescence molecule to it, a label that can tell you that every time your probe binds to this gene, you get fluorescence, and you can use it with different experiments. And DNA probes, we use them to experiments I'm going to talk about is an experiment called fluorescence in situ hybridization. We abbreviate it as FISH, and DNA microarrays. And this is where we create probes. And this is the same idea with the coronavirus. So if you sequence the RNA molecule and then you notice that there is a specific gene that you're interested in learning more about, you would create a probe that matches that RNA molecule, and you could focus on studying that gene. Maybe if you delete that gene in the virus won't be as infectious, or maybe you could use that gene to create a new vaccine that targets it. So FISH. So FISH is an experiment that uses DNA probes. And again, it stands for fluorescence in situ hybridization. In situ means computer. So in science, we use different types of terms. So we say in vivo. So if you're doing an experiment in vivo means in life. Those are usually animal experiments. In vitro means in lab, in a petri dish, and in situ means using a computer. So fluorescence in situ hybridization is you do these experiments and then use a computer to look at them. So we use this on non-cultured organisms typically. So remember, culturing organisms means when you grow organisms in lab, there's a lot of bacteria. I'm going to focus on bacteria right now. There's a lot of bacteria that we cannot get to grow in lab, but we still want to understand more about them. So we can do fluorescence in situ hybridization. You can create a probe that targets a specific gene, and maybe every time it binds to it, you get fluorescence. So it's used to identify both presence and location of a region of DNA using a probe. Presence, so you're checking, does your organism actually have that gene? So maybe you created a probe, again, that targets flagella, and you don't know if your bacteria actually has it. So you mix them together, and if you get fluorescence, it means that it's actually present because the probe binded the specific DNA, and it can also tell you a location. So I put a picture here from my own research when I was a grad student. And here I was trying to see if I was creating probes that target different genes for chemotaxis. Chemotaxis is just a fancy way of saying that the bacteria I was working with, H. pylori, can detect different beneficial chemicals in the stomach and move towards them, and it can detect chemicals that are not beneficial, that are harmful, and move away from them, which this is really good for the bacteria. It's not great for us. So I was trying to understand these genes a little bit more, and specifically the proteins that these genes code for. So here, for example, I was looking at a protein called Ki, and I added a probe, and every time you see fluorescence, it means that it bound to it. So it gave me an idea of here, the probe bound to the poles of the cell. So this gives me an idea that maybe these proteins are near the flagella and they control the flagella. And also we use fish to sometimes detect trisomy 21. Trisomy 21, as some of you may know, is Down syndrome. So at chromosome 21, you are supposed to have 2 chromosomes. Some individuals have 3 chromosomes, so that's an extra set of genes. That's why we call Down syndrome trisomy 21. And so you can create probes that target that DNA, and if you see fluorescence, you know that the organism, in this case, the human, will potentially have trisomy 21 or Down syndrome. Okay, we also use DNA probes in an experiment called DNA microarrays. DNA microarrays are very popular in science, and it's a method used to detect at many genes at one time using many DNA probes. So you use DNA probes you're interested in, checking different genes, and you put them on a glass or a chip, and then you add the sample, your cell sample, your bacterial samples, and you see wherever you get, usually it's some sort of fluorescence, that means those genes are on. So you can see which genes are on and which are off, and that's gene expression. So you can measure the level of gene products. You can see, does a cell create more of this gene, less of this gene? Maybe when a bacteria is stressed, it creates a lot of genes that will help it to create dormant cells in the future. So we use microarrays to look at level of gene products, so gene expression. Previously, I talked about fish. We use the fish to look at the presence and location of certain genes. With microarrays, we're looking at the level. So the fluorescent target sequences bind to the DNA probe and generate a signal, which is usually fluorescence. So for example, if you look at this plate, you can have a microarray, a chip that has probes that target different genes, and then you can compare a tumor sample with a healthy normal sample and see what genes are on or off in the tumor sample versus a normal sample. And it's not just on or off, it's more on, more off. It's levels. Like maybe I see this gene on 10 times more versus 2 times more in one sample versus the next. And this is important for creating drugs and a lot of other things. So that's DNA microarrays. Okay, DNA fingerprinting. DNA fingerprinting is a method that we use to figure out how to fingerprint people and organisms and figure out who's who and who's what. And this is where your DNA becomes important. So it's isolating and visualizing DNA to help identify an unknown, such as an unknown pathogen. So maybe all of a sudden you have people getting sick, so you want to know what bacteria is causing them to get sick, or what type of mold species. It could be anything. You can identify people who is the father in who is a father, who is a criminal. You can identify genetic mutations. You can do so much. And we use DNA fingerprinting in RFLPs, which are restriction fragment length polymorphism experiments, and PCR, which is the polymerase chain reaction. Now, I'm going to discuss both, but I want everyone to know that RFLPs are rarely used in science these days. We've advanced so much that we don't use it these days. But I think it's important for everyone to know, to understand the biological mechanisms, because if you understand how RFLP works, you can understand a lot of other experiments. So RFLP, what is restriction fragment length polymorphism experiments? So when we look at organisms, they each produce a unique pattern of DNA restriction fragments. So this is why the experiment is called restriction fragment length polymorphism. If you take an organism's DNA or a bacteria's DNA, they're different DNA sequences. If you add something called a restriction enzyme, restriction enzymes are enzymes that cut. That's all they are. So they cut DNA at specific sequences. So if I have one bacteria here and another bacteria here, they have different DNA sequences. If I add restriction enzymes to this organism and restriction enzymes to this organism, and the restriction enzymes cut DNA at specific sequences, I'm going to see different lengths, different fragments in this organism than this organism. And then you can compare those DNA fragments to known fragments and figure out, oh, this was E. coli and this organism was streptococcus that causes strep throat, for example. And we can visualize DNA using electrophoresis, which is basically the way that after we've cut the DNA, how we visualize the different fragments. So with RFLPs, you can identify paternity, who is the father. You can identify the strain of an unknown pathogen. This table is just a table telling you just different types of restriction enzymes. So for example, there's a restriction enzyme called BAMH or E. coli, and they cut at these sequences. So a long time ago, professors used to make students memorize what restriction enzyme cut where, but we know that everyone can look this up. So if you have a DNA sequence and you pipette on it, the restriction enzyme BAMH1, for example, it will cut everywhere there's GGATCC, it'll cut there. So every restriction enzyme has different places where it will cut DNA. And then once you've cut the DNA, you can run it on a gel. That's what we say in science, using gel electrophoresis, and you can look at the different DNA fragments. The heavy DNA fragments are at the top, the smaller ones are at the bottom, and you can see what do they match. So maybe this right here is my unknown organism that I cut, and then I compare it. Let's say these are all known organisms. I can see what it matches to see what that organism is. So once we've taken the DNA from the organism, we're unsure what it is, or if we don't know the father, and we add restriction enzymes to it, we run it on a gel, and electrophoresis separates DNA sequences by size. So it's kind of using electricity to separate DNA based on DNA's charge. Heavy DNA fragments, the bigger ones, will stay at the top. Smaller ones will run faster. And then after you're done with gel electrophoresis, you stain it with a stain that binds DNA. The specific stain we use most of the time is ethidium bromide. And then that fluoresces under UV light, so you can look at your fragments. So here are pictures of, for example, gel electrophoresis. So here in this sample, DNA fingerprints were used to track an infectious disease. So there was an outbreak of infectious disease in apple juice. So let's use this example. children in a school were drinking apple juice and everyone was getting diarrhea. So we knew that the juice was contaminated with an organism, but you want to understand what is the organism that's contaminated with apple juice, because there's so many different bacteria that can cause diarrhea, as an example. So what you do is you get isolates or samples from patients who drink the contaminated juice, and you get it from patients whose infections were not juice related or other different infections, and then you get the apple juice isolates and you see where the bands match to see where the infection came from. So did the infection come from the apple juice? Did it come from another source? And then you could also take the apple juice isolates and compare it to known organisms after you figure out where the infection came from. Another example, and I think this is an easier example to understand, so paternity tests, so who is the father? So you can take the mother's DNA, so if you have the mother's DNA, and then you have the child's DNA, you can add restriction enzymes to each. And remember, restriction enzymes cut DNA at specific sequences. So you take the mom's DNA and you cut it up with restriction enzyme. You take the child's DNA and you cut it up with restriction enzymes. And then you do gel electrophoresis. So here's the mom's DNA. Here's the child's DNA. And let's say you have 3 potential fathers and you're not sure who is the father. You take all 3 potential fathers and you take their DNA, you add restriction enzymes, the same restriction enzymes you used with the mom and the child, and then you run it on gel electrophoresis and you figure out this child, okay, this child, we know that this is the mom, so some of these bands are going to match with the mom, as you see here. Now you're going to figure out the other bands, who do they match with, and from me quickly glancing at it, I see that this child matches a lot with male 2, so most likely male 2 is the father. Okay, now we're going to talk about PCR. So we were talking about restriction fragment length polymorphisms, or RFLP experiments. Now we're going to talk about PCR. PCR stands for polymerase chain reaction, and I just want to tell you before I discuss it, polymerase chain reaction revolutionized the field of biology. So when this scientist came up with it, it changed our whole field, and we use PCR every single day. And PCR actually is one experiment that's being used in coronavirus testing kits. So I know we have the coronavirus outbreak right now, and I want everyone to know that a lot of these testing kits that are testing if a person does or doesn't have coronavirus infection with COVID-19 are using PCR. So PCR is the process of increasing small quantities of DNA for analysis. So it's specifically to amplify DNA, but you can look at specific DNA sequences. So for example, if we know that the coronavirus has a specific sequence, and it's an RNA virus. So not to get into the detail of it, you take the RNA from the virus and you convert it to complementary DNA, and then you use that DNA in different experiments. If we want to see, does the person have that complementary DNA from the virus's RNA, you can do a type of PCR, and what you do is you design these specific DNA probes. And remember, I told you that probes are DNA molecules that are complementary to another DNA molecule that you're interested in studying. And in PCR, we call these primers. So if you have a gene of interest here, you design a forward primer that binds this way and a reverse primer that binds this way so that you can amplify this gene. And the goal is to amplify this gene a lot. So that's what I'm going to talk about, the coronavirus example later of where you can use this test. So once you amplify the gene using PCR, you can visualize it on a gel. So we need to make copies of a gene for many reasons. If we want to study it more, if we want to do different experiments with it, if we want to actually insert that DNA in another organism. So this is where PCR is important. It's a method we use to amplify a DNA sequence. That's what PCR is. It's to amplify a DNA sequence, and you use primers and something called a thermal cycler. But the definition of PCR, it's the process of increasing or amplifying a specific DNA sequence. So maybe you have patient A, you're seeing if they have a gene and you're not sure if patient B has it. So you add these probes to see if you will detect that gene, because maybe that gene codes for a type of cancer and you want to see, do they have it? So what you do is in your tube, you have the patient's DNA. You're going to insert the primers, which are the fancy term for probes, that will amplify this gene. And if you see it in patient A and not patient B, you know that patient A has the DNA sequence, the gene, and patient B doesn't. And the thicker the band is, the more DNA. So polymerase chain reaction, you can visualize amplified DNA on a gel, and it's used for many different diagnostic tests for genetic diseases and detecting pathogens. Like I said, we use an alternate version of PCR that's a little bit more fancy for detecting coronavirus COVID-19 right now with outbreak. That's what most of these kits are doing. They're using some version of PCR where scientists have designed probes, or what we call primers in PCR, that will bind specific genes that we see in the COVID-19 virus. And if you see a band on an electrophoresis gel, you know that the patient has the infection. And if you see no band, you know that they don't. So here are, for example, Campylobacter species. This is a type of bacteria that causes very bad diarrhea, causes food poisoning typically when people eat contaminated chicken. So you can look and you can compare like Campylobacter species between two different organisms. There's Campylobacter jejuni and Campylobacter coli, and you can see the bands that are present in each that distinguish this species from this species. So we can distinguish specific species of bacteria. Here on this gel picture, can we call these gel electrophoresis pictures or gel pictures? There are individuals in the world, very few that we know of, that are resistant to the HIV virus. And you can be resistant for many different ways, but one of the main ways that you can be resistant to HIV is for HIV to infect, it has to actually get into your immune system cells, because that's what it specifically infects. So individuals who are resistant to this infection, the virus cannot get in because they have a mutation in the receptor that lets the virus in. So therefore, the virus cannot get in. If the receptor, remember, cells have protein receptors on them. If the cell cannot bind and get in, it can't infect them. So here we're looking at, do different patients have this mutation? So we look for this mutation on this gene, the CCR5 receptor, that's what it's called. And so here you can look at the gel and you can see wild type, wild type. Wild type is the most common. So this is what most people are, humans. We don't have this mutation. And then if you look at this individual, they do have this mutation. So they have an extra piece of DNA here that we've amplified using PCR. Okay, so that was PCR, and you can use PCR for many different things, but the gist of it is it's used to amplify DNA on a gel, and you can actually specifically see, does an organism have that DNA, by composing PCR primers that will bind to that DNA. DNA cloning, another experiment we're going to learn about, and the rest of this lecture is going to be about how do you clone DNA in an organism. So DNA cloning, it's a fancy term for saying a procedure where a gene of interest is taken from an organism and inserted into another cell, where you can get that cell to express it and make it for various reasons. So I'm going to use this example. You can do DNA cloning in so many different ways, but let's say that, okay, so we talked about this example a lot in class. Let's say that you have an individual who's lactose intolerant, meaning that they cannot digest lactose, most likely because they are missing the lactase gene. So lactase is the enzyme, the protein that breaks the lactose sugar. So usually most people who are lactose intolerant don't have or don't make enough lactase. So let's say that you're going to be this new company that's going to create lactase, the enzyme, and you're going to give it to people who are lactose intolerant. Now, you're going to take the gene that codes for lactase from a healthy human that makes that gene. So here, if we look at this image, let's say we get the human genome. The human genome is all the DNA, and you're specifically studying the lactase gene. You don't care about all the other genes. You don't care about the genes that make the person move or help their heart or help with many other functions. So what you're going to do is you're going to isolate that gene from the person, usually using PCR, where you just get the lactase gene. After you've isolated just that gene, you're going to put it on a bacterial plasmid. We're going to talk about how this is done. And then you're going to take this bacterial plasmid and you're going to stick it back into bacteria. And we're going to stick into bacteria because bacteria reproduce really quickly because they just divide by binary fission. And once you get the bacteria to make a lot of your gene for you, maybe you'll open up the cell and extract the protein. Because remember, genes are transcribed to mRNA and they're translated to protein, and lactase is an enzyme, so it's a protein. So this is all an example of DNA cloning. So DNA cloning involves recombinant DNA. Remember, it involves recombinant DNA because recombinant DNA is when you have an organism that has DNA from 2 or more sources, a combination of DNA. And genetic engineering is basically, gene cloning is a form of genetic engineering where you insert genes into cells, which involves recombinant DNA. Recombinant DNA is an example of any DNA piece that has DNA from 2 or more different sources. So, for example, you can have recombinant DNA. You can insert human insulin gene into bacteria to get bacteria to make you insulin, and then mass manufacture it to give to people who need it. You can insert virus envelope genes into yeast organisms, have the yeast make it for you. And the reason we would put it in yeast is a eukaryotic organism, and then possibly produce vaccines. This could be potentially what they're doing with the coronavirus vaccine development, looking at what genes trigger an immune response in humans. Can we take pieces of those genes and use them in vaccine development to have people create an immune response to that? And we use bacteria and yeast as where we do most of our cloning because they're mass producers. They reproduce really quickly, and each of them has their own benefits. And the genetically engineered organisms are called recombinant organisms. So your genetically engineered bacteria that has an insulin gene from humans is a recombinant organism. Your yeast organism that has a gene from some virus is also a recombinant organism. And we clone a lot of human and plant genes and bacterial organisms, again, because bacteria reproduce really quickly by binary fission, and they make a lot of copies or clones of that DNA for us. This is why we call this process DNA cloning. You have a specific piece of DNA or a gene, because remember, genes are DNA, and you're trying to make a lot of copies, whether that's for mass manufacturing or to create a vaccine. So you're cloning it. So this is why this process is called DNA cloning. And the product that you're creating could be ultimately a protein or just the DNA sequence that you're interested in. So now we're going to talk about how cloning is done in lab. I've done a lot of cloning while I was getting my degree in graduate school. So cloning is very popular. A lot of companies hire students to do DNA cloning. It's a very great method to understand how to do. And you guys will hear about it a lot in your lifetime. You'll see a lot of jobs that are hiring people to specifically do DNA cloning. So the way you clone a gene into an organism, so let me actually put them all out. Okay, the first thing is you want to obtain your DNA for your gene of interest. So you want to get the DNA, and then you want to get this specific gene that you're interested in. You want to cut the DNA at restriction sites, and you want to cut the plasmid that you're putting it in. And then you combine your cut piece of DNA with your cut plasmid. And to make this lecture easier, I'm going to say a vector and a plasmid are the same thing. So your textbook will say vector. Just know that for purposes of this explanation, a vector and a plasmid is the same thing. So now you have your new recombinant DNA plasmids or vectors that you've made, and now you want to insert them into the host cell. And then once you have them in a host cell, your bacteria, you want to make sure the bacteria actually took in that recombinant DNA. So you do a few experiments to make sure they actually have it, and then you can collect your final product. So obtaining the DNA for your gene of interest. So the way you do it, we're going to talk about. So the first step is obtain the DNA for your gene of interest. You need to get your DNA. So let's use our same lactase example. You're trying to manufacture lactase. So the first thing is you need to get your lactase DNA, for example. So you're going to lyse the cells. You're going to take your human cells. You're going to lyse them because you want to get to the DNA, and you're going to get the DNA. And you're going to cut the DNA at specific restriction sites. And the reason you do this is you want to cut the DNA so that it can match your plasmid or vector and they can complementary bind to each other. And you use restriction enzymes to cut the sequences of DNA, and you create these ends called sticky ends, and they're complementary to each other because you use the same restriction enzymes. And then the plasmid and your gene of interest will complementary hydrogen bond to each other because A will be attracted to T's and C's will be attracted to G's. So the DNA fragments can hybridize, which means stick to other DNAs that are cut with the same restriction enzyme, and because DNA forms hydrogen bonds with each other. And then after you have your cut DNA fragment, you need to insert it into a vector or a plasmid. So you cut that plasmid with the same restriction fragments to create sticky ends, and then you can get them to stick to each other. So a vector, the definition of it is a carrier piece of DNA. Vectors can be anything. Most vectors are plasmids, so they have to be able to self-replicate. Let's just make it easy and say all the vectors right now are plasmids that we're using. And we use plasmids because they can self-replicate. So now you made your recombinant DNA plasmid or vector. Now you want to insert it into host cells. And usually the host cell you're going to try to use is something that reproduces relatively quickly, is simple to work with in lab. So a lot of times what we take our recombinant DNA molecule or DNA plasmid that we made and we stick it in E. coli or Bacillus subtilis because these bacteria are easy to grow in lab. They've been well studied. And the way, so you have your DNA that you made, your recombinant DNA, and you have your, let's say your bacteria, and you want to get this DNA in the bacteria, you have to figure out a way to get that plasmid in the bacteria. The way we do this is through transformation, specifically artificial transformation because it's being done in lab to put the plasmid in the host. You need to take the bacteria and somehow compromise its cell membrane and cell wall so that it can take up the DNA, but you don't want to kill the bacteria. So two examples of being able to do that is you can heat shock the host, the bacteria. Heat shock is basically taking the bacteria, sticking them in warm water, then ice, then warm water, then ice. And this makes the plasma membrane a little bit fluid, and then they can take up the plasmid. Or you can electrocute the hosts, the bacteria. You barely electrocute them. If you electrocute them too much, you'll kill them. I've definitely done this in lab many times during my own research. You do not want to kill the cells. You just want to barely make the cell membrane and cell wall of bacteria a little bit more fluid so they can take up your plasmid. And these methods are called artificial transformation. And then once you get your recombinant DNA in the bacteria, you're going to culture the bacteria. Culture, again, is a fancy word for actually grow the bacteria. So once you grow the bacteria, you hope that they took up your recombinant DNA, but you want to make sure that they actually have your gene of interest. So you have to confirm that they have your recombinant plasmid. So what you can do is you can do blue-white screening. Blue-white screening is a way to look for recombinant plasmid. So again, not all the cells are going to have taken your recombinant DNA, and we're only interested in the cells that took it up. So we do an experiment called blue-white screening, and I will talk about that in a second. So blue-white screening, there's specific plasmids that we use in transformation, and when you look at these plasmids, they have an ampicillin-resistant gene, and they have a LAC-Z gene. LAC-Z codes for beta-galactosidase, and beta-galactosidase, what it does is it breaks down a certain chemical and produces a blue color. So if your bacteria have your recombinant plasmid. Your recombinant plasmid has lac-Z, and if you grow that bacteria on an X-Gal plate or an agar plate that has the chemical X-Gal, this lac-Z will break it, and every time it breaks it, you get a blue color. So that way you're sure that your bacteria took up your recombinant plasmid. That's just one example. I didn't want to get too much into the detail of it, but another way you can make sure that your bacteria took up your recombinant plasmid is some of the plasmids have an ampicillin resistant gene. So what you do is after you grow the bacteria, you plate them on agar plates that have the antibiotic ampicillin, and only the bacteria that have resistance to ampicillin will grow. So that way you're selecting for bacteria that are resistant to ampicillin, therefore have the recombinant plasmid that you created with your gene of interest. And usually your gene of interest is somewhere here, where the restriction fragment, where the restriction enzymes are. So ways that we can make sure that our bacteria took in the recombinant plasmid is you can do blue-white screening, which is what I described. You can also look for the specific gene of interest. So maybe you can do DNA sequencing to make sure did your bacteria actually get that gene. You can see, remember, genes are transcribed to RNA, and RNA is translated to protein. So ultimately genes make protein. You can see, did your cells actually make lactase, the protein? And you can do colony blotting. I didn't explain colony blotting in this lecture because no one uses it anymore, but it is an example of a method that I wanted to put on here. So finally, you collect your product. So after you get your bacteria, you insert your recombinant plasmid, you made sure that they grew, you make sure that they're making your product, you need to actually collect your product. So in our example, if we made lactase is a protein. So what you would do is you would grow a lot of the bacteria, you would lyse open the cells, and you would get the lactase protein. So you do protein purification experiments, and you would just collect the product and do whatever you need with it. Okay, so just to recap this whole thing of how it's done. So these are the steps that we talked about. Okay, so we're going to go through this picture just to talk about it again. So what you're going to do with cloning a gene is you're going to isolate plasmid DNA from your bacteria, and you're going to get your human DNA of interest. You're going to cut the gene that you're interested in, or the DNA from the human, and the plasmid with the same restriction enzymes. Once you cut them with the same restriction enzymes, they'll have extra bases that we call sticky ends. When you put them together, they'll hybridize, so the plasmid will stick to the DNA fragment. Now you have your recombinant DNA plasmid. The next thing you need to do is actually to get that plasmid that you made into the bacterial cells. So you could do that through artificial transformation. You could either heat shock cells or do electroporation, both types of artificial transformation. Once you have your recombinant plasmids in the bacteria, you need to grow the bacteria and screen to make sure that they actually got your recombinant plasmid or the recombinant DNA. One way to do that is through blue-white screening. So you put the bacteria on agar that has ampicillin and X-Gal, and you make sure that they grew and they're actually blue colonies, because your recombinant DNA plasmid has the X-Gal gene. I apologize, has the lac-Z gene, which will cut X-Gal in the agar plate, resulting in a blue color. And then finally, once you make sure that you have those bacterial colonies, you'll grow them and collect your final product. There are a little bit of differences in getting genes from eukaryotic organisms to prokaryotes, etc. So if we want to take eukaryotic genes and put them in prokaryotic hosts, for example, if you want to take the insulin gene from humans, a eukaryote, and put it in bacteria, a prokaryote, there's some extra things you have to do. So to put eukaryotic genes in a prokaryote, first cDNA must be made from the eukaryote. So what you're going to do is you're going to get DNA by reverse transcription. So let's look at this image here. So remember that in eukaryotes, when we look at a gene, it has exons and introns that are transcribed to RNA by RNA polymerase. Once you have your RNA, you need to do RNA processing, and you need to, remember, you need to remove the introns and put together the exons, because exons are expressed and introns are intervening sequences. Now you have your mRNA, and that mRNA actually codes for the protein you want. So when you want to work with humans, you're going to get the RNA molecule and you need to convert it to DNA to stick it into bacteria. So when you get your RNA, you need to convert it to DNA. So remember, DNA to RNA is transcription. If you want to go the other way around, RNA to DNA, that's called reverse transcription. And to convert an RNA molecule or an mRNA molecule to a DNA molecule, we use an enzyme called reverse transcriptase. And the DNA that's made, we call it complementary DNA or cDNA. And again, we do this because you cannot just take the eukaryotic DNA because it has a lot of intervening sequences. You don't want those intervening sequences. So eukaryotic mRNA is what you work with. So you would isolate the eukaryotic mRNA that codes for lactase, and then you convert that to DNA using reverse transcription, using the reverse transcriptase enzyme, and then you make your DNA, which we specifically call a complementary DNA, and then you use that complementary DNA to do gene cloning in bacteria. Now, if we want to take genes and put them in a eukaryotic host, now we're not talking about working with bacteria. We're talking about working with eukaryotes. What if you want to put a foreign gene in an animal, or you want to put a foreign gene in a plant? How do you do that? There are different ways. So to put genes into eukaryotic hosts, such as animals, plants, other eukaryotes, there are different methods that can be used. I'm just going to talk about a few. So one way is you can do artificial transformation. It's not as effective with eukaryotic cells from my knowledge, but you can also do two methods, which I want to talk about. So when we're working with plant cells, if we want to insert, let's say you want to insert a gene that makes watermelon seedless, so it deletes a seed gene, you will want to insert that gene into plant cells, watermelon cells. So one way to do it is through a gene gun. So DNA is brought into the plant cells by a DNA, we call it a gun, and it basically shoots a burst of helium into cells and plant cells and opens them up so that they can take up your DNA. So we use gene guns with plant cells. Animal cells, if you're trying to transform an animal cells with foreign DNA, you can do microinjection. Microinjection is using an injection, a tiny injection, to get your DNA into the animal cell. Remember, you don't want to harm the animal cell. This is why we use microinjection. Now, after we've learned about gene cloning, you can imagine that this is used every single day in multiple industries, medicine, plants, forensics, different types of industry. So we're going to talk about different useful products that you can get from gene cloning. So, okay, with gene cloning, you can make a lot of useful proteins and you can make a lot of useful DNA sequences. So let's talk about protein. You can make insulin. You can get bacteria to make you human insulin, and you can use diabetes comes in many forms, but most diabetes are treated with giving the person insulin. So insulin is a protein and it's been made through gene cloning. If you have someone that has a growth deficiency for whatever reason, you can give them growth hormones, which are proteins that are also made through gene cloning in bacteria. There's something called the factor VIII protein. It's an essential blood clotting protein, and patients who have hemophilia A, if you've ever heard of it, lack this protein. So you can give it to them through gene cloning. So you can get bacteria to make you that protein, and then you can present that as medication or treatment for people who have hemophilia A. Vaccines. So what vaccines are basically injecting components of a pathogen or the dead pathogen into an organism to get it to create an immune response, so that if you ever see that organism, you're not harmed by it, you don't get sick. So a lot of times what we do is we take pathogenic proteins, or proteins that actually make something a pathogen, that makes you sick, and we take those proteins and we use them to create vaccines. And then that vaccine is a protein subunit vaccine, so that when you get that protein from the pathogen, your body creates an immune response without you actually getting the disease. Commercial products. Gene cloning is used so much to make so many different things commercialized. So RBST, I think many of you guys have read milk that's like RBST free. So it's bovine growth hormone, and it increases milk production in cows. So a lot of farmers use this hormone and you get a lot of milk. You can imagine why this is beneficial if you're a farmer. There's a lot of talk of, does this also affect humans? That's why there's a lot of milk lately that tells you RBST free. So much research on this, and we can talk about the benefits and the harm of it anytime I see you guys, but this is one example where we've used it. Another is we use different proteins to make cheese, to get cheese to stick to each other, and to get different textures of cheese using protein. So that is another way that we make cheese. We can use gene cloning to make DNA sequences of interest. So maybe you have someone who has a mutated gene and you want to give them a gene that's not mutated, a healthy gene. DNA vaccines are still very experimental. We're trying to see if we can get DNA from pathogens and put it in a vaccine and use that to get an immune response from people. Okay, so if we were in class face-to-face, I would ask you guys, could this technology be used to create bad products? And the answer, definitely. You can create anything you want. You can create antibiotic resistance genes. You can create genes that make organisms grow out of control. So there's so much more benefit to gene cloning than harm, but I just want you to be aware of all the things we can use gene cloning for. And our synthetic designer babies in our future, possibly. We talked about CRISPR last time, and before CRISPR is, you can do so much gene editing and it's very simple, but before CRISPR, people were using gene cloning to see if they can create the perfect baby, whatever that is, by inserting genes and removing genes. So just keep that in mind. Transgenic organisms. We're going to talk about different organisms that have been made using gene cloning. So transgenic organism is when an organism has foreign DNA present in every single cell. Remember, multicellular organisms have multiple cells. So if we see foreign DNA from some other organism in every cell, we call that transgenic organism. For you guys and for what you guys probably have seen in the public or in the supermarket is GMOs. They're genetically modified organisms. So transgenic organisms are organisms that you've modified genetically and they have foreign DNA. How can you introduce foreign DNA into an organism? Gene cloning. This is what we learned about. So examples of transgenic organisms, we're going to talk about it. You can turn fruit different colors. So strawberries are typically red. If you insert a gene that makes them another color, that's a transgenic strawberry. It has the gene that now codes for blue color. You can create animals that fluoresce. Now, we actually do this in lab in medicine. I'm sure it's used in other things, but I'll tell you in medicine, maybe you're studying a specific protein and you're seeing where does a protein that causes disease and you're trying to see its location. So maybe you add fluorescence to that protein and you clone it into a mouse, and therefore you can see where the fluorescence is. You can get animals that are very big. You can get so many different things. In 1994, the first genetically engineered food went to the supermarket, and here's a tomato that's been genetically modified and it stays riper longer off the vine. So usually fruit and vegetables, as long as they're on the branch of the tree, they stay better longer, but the moment you remove them, they go bad. So we created transgenic tomatoes that stay riper and look better and last longer. You know what? I always tell this to my students. A lot of times when you go here to American supermarkets, like if you go to Safeway or any supermarket and you go to our fruit and vegetable section, everything looks so perfect. Like apples are perfect and tomatoes are perfect. And I know my grandma, again, who's from Syria, from another country, she always comments on how fruit looks so perfect here, but is kind of tasteless. If you go to a lot of different countries, when you go to the fruit area, you see fruit that does not look this perfect. Yeah, some of them do, but most fruit, there is bugs on it. It's changed color. It's become more ripe. This is what happens naturally to fruit. And the reason why our fruit looks so perfect when you go to the supermarket is because of so much genetic modification. And there is some questions if you actually genetically modify a fruit to stay looking really good, to stay very red and stay very plump and big, is it generally going to alter the taste if 2 weeks later the tomato still looks the same, but it doesn't taste as good? So there is a lot of talk about that. Transgenic organisms, so we're going to talk about where we've used them in plants and agricultural applications. So we've used gene cloning to increase shelf life of fruits and vegetables, make them last longer, keep them on the shelf, keep them looking perfect by I use the word playing, but altering different genes. Drought, salt, frost, chemical tolerance, so making your plants or your farm tolerant to salts, if you live somewhere where there is a lot of snow, can you get your plants to tolerate that snow? You can get plants to produce biofuels, different pharmaceuticals, enzymes, plastics, rubber. You can get plants that are helpful in capturing pollution and carbon to help with global warming. You can get crops that fix nitrogen. So nitrogen fixation is important because when we fix nitrogen, we can get more plants to grow. So if you get plants that can do this, you increase your crop. You can get plants that are allergen-free, low-maintenance lawns, caffeine-free coffee beans. So coffee naturally has caffeine. How you make caffeine-free coffee is you delete the gene that codes for caffeine. And edible vaccines. This is a big area of research of can we create edible vaccines. The benefit of creating edible vaccines, as you can imagine, is it can be accessible to everyone and you don't need healthcare. Potentially, you wouldn't need healthcare workers to administer them. So if you have countries that don't have a lot of money, maybe you can create edible vaccines. Pest resistance, you can add genes that make plants resistant to different pests. So there's the PT toxin gene, and it gives plants resistance against insects. Viral, bacterial, and fungal resistance genes too. So the Bt toxin gene is a very popular gene that we put in plants, and again, it makes them resistant to insects. Herbicide resistance against weeds, so glyphosate or Roundup, if you've ever seen this in the supermarket, if any of you are into planting, it gets the plants to be resistant against herbicides. And then improved nutritional value, you can make rice more nutritious, have more vitamins by inserting genes that create these vitamins. Canola oil with enhanced vitamin E or fatty acids, so get canola oil to create more vitamin E or fatty acids. Now, transgenic organisms. Now we're going to talk about organisms. We were talking about plants before. There's a lot of research on human diseases using animal models. So let's say that you are in a cancer lab and you're researching different cancer drugs. It would be beneficial to be able to test cancer drugs on organisms that have cancer. So one organism that's used in lab a lot are mice or different types of animals. So you can get, there's something called the oncomouse, and in science, and if you guys don't know, oncology is a field of cancer. So an oncologist studies cancer. The oncomouse is a mouse that has the gene that makes it susceptible for cancer research. So it's basically a cancer mouse, and it's been genetically modified to have a form of cancer. And the great thing about the oncomouse is if you're in a lab and you're researching different drugs for cancer, you can order a lot of oncomice and you can use them to study your drug. Fluorescent animals, the GFP gene. So green fluorescent protein is a protein that, from my knowledge, was isolated from jellyfish, and we use it to tag or to stick to different proteins in animals to see where those proteins are going. So if you look at this picture, maybe this lab was studying proteins that go to extremities, so to the ears, to the tip of the nose, to the legs, the tail, and it'll give you some idea of whatever it is protein you're studying. And then aqua advantage salmon. So I don't know if anyone knows this, but salmon is generally not supposed to grow year round. We've genetically modified fish so that they can grow year round, not just in the spring and summer, different types of fish. So again, that's a genetic modification where you've altered their genes. And another way is farming. So this is, I want everyone to know the definition of farming. It's using milk or egg producing animals to make pharmaceuticals. So using animals to make different pharmaceutical products. Atrin, antithrombin goat, inhibits blood clotting. So this is a medication that we've gotten from a goat, and it's a medication that we use in people who have blood clotting. So this is where you get it. Organ transplants. So I want everyone to know the definition of xenotransplantation. It's when you take non-human tissue and put it in a human recipient. This is like taking a kidney from a pig and putting it into a human. This is an example of a transgenic organism because you're putting two different organisms together. Disease control, we have the malaria-proof mosquito. The way we created malaria-proof mosquito is through altering the genetic sequence of the usual mosquito. Lysozyme in goat milk for diarrhea control. So there's this enzyme called lysozyme, and what it does is it helps with the different sugars and different components in goat milk so that some people do not get diarrhea. Environmental protection, we have the EnviroPig. It's a GMO pig, and it can digest phosphorus more efficiently, and that's good for the environment if animals are producing low phosphate feces. There you can see all the benefits of transgenic organisms, and I just talked about a few. There are so many more examples in plants and animals and bacteria of beneficial things we've done and are continuing to do for society using gene cloning, but there are a lot of concerns with transgenic organisms. Could you introduce allergens, toxins, carcinogens by doing all this gene cloning? Are you accidentally going to, by inserting a gene, make a toxin or make something that causes cancer? And changing ecology. Okay, if we're creating all these plants that are resistant to so many things that we would term bad, like weed or pests or anything like that, are we changing the ecology? Are we killing good insects? Are we harming plants? By killing good insects, if the insects have nowhere to feed, are we killing them? Did we need those insects? Are we creating plants that are all the same and there's no diversity? So if the world changes, everything is just adapted to what we created, and escaping and outcompeting wild counterparts. We're losing natural selection by doing this. So you really do not want to change diversity. And gene therapy in people. So this is another final topic that we're going to talk about in this lecture because it has to do with genetics. Gene therapy in people is a form of gene cloning. And let me put it all out and we'll talk about it. So it's still very experimental. It's not licensed yet at all. It could potentially be licensed in the next few years, but it's not. And it's gene cloning in people to replace defective or missing genes. So if a human is lacking a gene that's important, can you replace it? Or give them the gene that they're missing. So future hopes are, can we use gene cloning to cure cystic fibrosis, sickle cell anemia, hemophilia, other cancers? So cystic fibrosis affects your lungs, sickle cell anemia affects red blood cells, hemophilia also affects blood, and cancer. Can we treat those using gene cloning? Now, I do want to say right now with this slide, gene cloning has been a big field, but now with our advance in CRISPR, CRISPR might be taking over. So just to keep that in mind. And with gene therapy, the idea here in the US is that only dysfunctional cells would be genetically altered. So you cannot change genes just for someone's benefit. It's only if someone has a disease and it's replacing a dysfunctional gene with a normal gene. So there is a disorder or a disease called SCID, or severe combined immunodeficiency, where humans are missing almost all the genes that code for a healthy immune system. We'll learn about it later. Can we give those humans those genes back? And alter functioning of cell genetics. So can you change skin cancer and blood cancer so that those cells are healthy again? Can you alter HIV? So in this example, we're basically giving a gene to a human that's missing that gene. So you guys can see this example. And then here are the study questions, the practice test. So I think the video is done from here, and you guys can email me if you have questions. Okay.